Hi, it's Brett, the lab tech here. Sorry for the noise. We're outside today because we're going to be doing a photosynthesis experiment. So, what we did last week is we took one of these geraniums and we set it up with filters that look something like this. So we have blue, red, and green, and then a place on the leaf that is covered by a black piece of tape. So that's going to be our experimental setup. And we're going to be looking for the presence of starch underneath each of these areas, and then we can also compare it to the unfiltered areas. But before we can see the starch, we have to get all this green pigment out of the way. And to do that, we need to boil the leaf in alcohol. And because I don't have a fume hood inside my house, we are going to do this out on the front porch, so to get adequate ventilation. So the first thing I'm going to do is put on some personal protective equipment. Put on some goggles. I don't want boiling ethanol splashing in my eyes. That tends to ruin my day. And I don't want to go to the hospital right now. The next thing we're going to do, so down here we have a hot plate. Actually, I'm going to get a little further away from the siding, just in case anything goes bad. We have some tap water in a 1,000 milliliter beaker. We've got about 200 mils. It doesn't matter exactly. You just don't want so much that the other container can't get in there. Actually, I'm going to take the label off. Might melt when this gets hot. Then the other thing we have is 80% ethanol. This basically, we're going to make a double boiler since we don't want to heat the ethanol directly on the hot plate. And then we will turn this all the way up. All right, so it looks like the ethanol is getting up to temperature nicely, so I think we're good to go ahead and put our leaf in. So we're going to take a leaf off the plant, and before we boil it, we want to keep careful track of where all these filters are. So go ahead and sketch this out in your lab manual right now. And the other thing I'm going to do, just so we can keep track of how this filter was oriented, I'm going to put a little punch hole in this leaf. So now we can keep track of where all these filters were. Okay. So now that we have everything kind of located on this leaf, I'm going to go ahead and put this into the hot plate, or not into the hot plate, into the alcohol that's on the hot plate. And actually it's getting a little warm, so I'm going to turn it down because we don't want this boiling violently. Ideally, we would have some boiling chips in here, but I don't happen to have any boiling chips. So we're just going to do this carefully and keep a close eye on it. So this is the leaf before we put it into the boiling water. And you might even see that there are some areas of this leaf that are a little bit lighter, that don't have quite that dark green pigment on it. So, and the filter was on there, something like this. So we're going to go ahead and put the leaf into the alcohol and let the pigments extract out from the leaf into the alcohol for a little while. I'm going to turn this up. And again, you don't need to wait with me for this whole process. Actually, I'm going to move the camera so you can kind of see what's going on inside. Okay, we will pause the recording for a while. Okay, looks like our leaf is boiling nicely. We can see that the water isn't really boiling anymore, but the alcohol is, because alcohol has a much lower boiling point than water. And I'm going to carefully get this leaf out, and we can see that it is pretty much entirely bleached. So it could maybe go a little bit longer. I'll put it back in just briefly. I'm going to show you, this is the leaf. It is entirely done now. So we're going to put that in our little tray here. The other thing I'm going to do is rinse it off with just some distilled or deionized water. Just kind of rinse the alcohol off, cool it down a little bit. And then the other thing I want to do is make sure to turn the hot plate off. And I'm going to unplug the hot plate and also take the beakers off the hot plate. And let them cool down, let everything cool down for a while. So here we have our boiled in ethanol leaf. The pigment has been almost totally bleached out of it. I'm gonna put the rinse water in the container. There might be a little ethanol in there. You do wanna make sure, if you do this in the lab, to make sure to put all the ethanol in the appropriate waste container. I'm going to add maybe just a little bit more water and kind of put this leaf up very gently. The leaf is very fragile after boiling. That looks pretty good. And then now we are going to add Lugol's iodine. So this is going to stain for starch. Lugol's, when it meets starch, makes a dark blue or kind of purplish black color. And this is going to tell us where the starch is inside of this leaf. So we're going to get about 4 or 5 droppers full. And let it sit. For a little while. It looks like this is staining very, very quickly. And actually, I think that's probably as dark as we want to let that get. So I'm going to rinse the Lugol's off and stain it. Add some rinse water to kind of stop it from staining even further. Okay. Now we are ready to take a look at this. And to do this, we want to orient this the same way that it was on the plant. I'm going to turn the leaf over. And it was something like that on the plant. And then just as a reminder of where all the filters is, or where all the filters were. So we had that punch hole. Hopefully you can see that. And then that dark spot where the black tape was. And then we had our 3 different colors that were kind of arranged like that. So blue, red, and green. Okay, we've got a light on here. So hopefully we can see the results a little bit better. And that is what it looks like. A little more spread it out. Yeah, so again, blue, red, green, and then black. Okay, so that is the photosynthesis experiment. Thanks for watching.